Okay.
All right. So I have kind of a long title because there's like a few things I have to focus on here that like aren't kind of sequential, I guess. But I'm getting a little more into RAS and the differences in structures between its GTP and GDP bound states, the mutations, one specific that I'll be talking about, and ways to bind to that specific mutant in order to kind of inhibit it. So as an introduction, I went through two papers. The first one is titled KRAS G12C, which is the mutant, inhibitors allosterically controlled GTP affinity and effector interactions. And essentially just the breakdown is it's targeting an allosteric site in RAS, which means it's not the binding site of GTP or GDP. It's a different site on the protein that causes a conformational change that can inhibit it. And it's identifying and testing these cysteine reactive small molecules. And that's important because G12C means it's changing a glycine to a cysteine. And cysteine reactivity is something that we know a lot about. And then the second one was actually the 8th reference in that paper, which we'll talk a little bit more about in the end of the presentation. And it's the tethering method for identifying cysteine reactive small molecules. So this is how they actually went about the experiment in the first paper. And then in the presentation, first I'll go over the differences in structures between GTP and GDP-bound RAS, because there's two switches that are very important, the description of those two switches, and then the KRAS G12C mutation and this new allosteric target pocket, and then the tethering strategy for how they found these small molecules to target it. So when RAS is bound to GTP, it is activated, and when it is bound to GDP, it is deactivated. So this is just the same schematic that I had in my previous presentation, just as like a refresher. But there's a key difference in the structures between activated and deactivated RAS, which is why they allow to bind to these GTP or GDP. So here are the structures I put in Chimera. So this is GDP bound, which is off, GTP bound, which is on. So I'll point out a few key things. These green dots, like I said, are the magnesium 2+ ions that are complex within the protein. As you can see, this one is kind of complex to 2 sites, and this one is complex to 4. We'll get into that a little bit later because of the switch in the protein that allows that. You can see the 2 phosphate groups here, 1, 2, 3 here, and there's a few subtle differences that you can notice before I actually get into the switches themselves. If you see this helix right here is kind of shifted upward in the GTP bound, so it's more of a loop in this upper corner here. Now it's more of a helix. And then this loop is closer to the magnesium ion here. That's why it's able to be complex to the specific position. And this one is further out. So those are just a general description of the main differences between these proteins. And now here's like the full structure. So you can see how the nucleotide fits in. And then you can still see these subtle differences, how this part of the protein actually shoots more upwards. And this is a little more condensed. So that is what I'll get into in this next slide. So these are called switch 1 and switch 2. So most of the structure of RAS doesn't actually change. So as you can see from this little image, the main body of the RAS protein isn't affected, whether or not it's bound to GTP or GDP. But there's these 2 particular amino acids that are bound via hydrogen bonds to the gamma phosphate of GTP, which just means the last one. So alpha, beta, gamma, I think. And then this changes the orientation when RAS binds to GTP. So it's like a loaded spring mechanism, which is how I was going to describe it. So when these 2, the glycine 60 and the threonine 35, when they're bound to that gamma phosphate, they're inwards. And when that phosphate is cleaved, essentially, when it's switched out to GDP, they spring open, which is why the 2 structures look so different. And if you go back to here, that little stick looking thing right there that the magnesium is complex to, that's the threonine 35. That's moved closer inward to that 3rd phosphate, so it's able to hydrogen bond. So here's just a better picture of what the switches look like. So I kept the same color scheme. Dark blue is GTP and the teal is GDP, and I just overlaid them. So both switches occur in what's known as the loops of the protein. So they're between helices and sheets. So if you look at switch 1, which is up here, you can see how these 2 loops do not overline with each other. And the reason being is here's that threonine 35, and there's the 3rd phosphate. So it's able to bond to it, so it's pulled closer in than this blue loop right there. And then switch 2 also occurs in the loop, which because of this causes the like non-overlap of those two helices on the outside. And I can't actually highlight just one glycine in the structure, because if you go on Chimera, it'll like highlight all of them, which made it look really messy. But it's somewhere in here, in this interior portion of the dark blue loop, so it's being pulled more in towards that third phosphate.
I can show you how to do that, Beth.
Oh, there is a way?
Yeah,
okay, we tried it for like an hour.
We can talk about that later. Sorry about that.
Cool.
Okay.
But if that makes sense, this is the differences between these two structures. It's just the threonine 35 was luckily for me highlighted a little bit by showing it off the loop of the protein, so you can actually see that it's moved more inward than the light loops. So now what's interesting is this mutation also causes changes in the protein itself. So like I mentioned, G12C is just a notation for indicating that the glycine at position 12 in the amino acid sequence of the protein is now a cysteine. So this is called a point mutation, which leads to a missense mutation. So a point mutation is in the DNA itself. The missense mutation happens during translation. So one base pair, either the first or the second, because the third is called the wobble base pair. So if it changes, sometimes it doesn't actually change the amino acid sequence, was switched, causing the codon, that 3 base pair sequence, to be read as a different amino acid, causing it to switch to cysteine. This mutation, why it's dangerous, is it keeps RAS activated. So it's constantly transmitting that signal downstream, allowing for more cell proliferation, which causes cancer. But what's interesting is because of this 1-point mutation, there's a new little pocket next to the cysteine and between that and switch 2, which I'll show in a second, which is now a target. So because of this mutation, we found a new way to actually inhibit the protein. And the switch 2 is reordered, so it's a little messed up because of the mutant, but switch 1 remains like unchanged. So if you, as you'll see in the structure, switch 2 looks a bit wacky, but we now have a new way of targeting the enzyme. So here is like the ribbon form, and I was able to highlight one cysteine, and I don't know how I did it, but it's in yellow. So there's that magnesium ion. This is obviously the GDP bound form. So there's that just one GDP. This dotted line is where switch 2 is supposed to be. So if you remember from the previous structure, that switch 2 was like kind of hovering right over the magnesium and included part of the helix. Now it's like really disordered, so they indicated it as a dotted line. And this PDB came directly from the paper, so I hope it's right. But now you can see that that cysteine is right near the pocket where GDP binds. And because of that, there's this new indentation right here that they are able to target. So the pocket contains these 2 zones, essentially, that they are able to work with. And they're very deep. It's hard to see depth within these structures itself, but it allows for these hydrophobic interactions because it's so far into the protein itself, which helps for designing small molecules. So this is how they actually did it. So the tethering approach came from a reference in the paper published in 2000. And fun fact, both papers actually came from people at UCSF. So good things coming from there. But this is a method of identifying these small ligands that bond loosely, which is interesting, to the target site in the mutated KRAS via a tether. So it's somewhat similar to Naya's PROTAC approach, which has a linker between these two things to get the 2 proteins to kind of come together. This is a tether between 2 positions in one protein. So this is a picture from that paper. And so there's that cysteine. It's the only amino acid with a thiol group. And you like kind of bombard it with all these small molecules that have disulfide bonds, which are easy to form and break. And then 2-mercaptoethanol is just a disulfide bond reducing agent. And so you see, if this little small molecule can bind to the protein and form a new disulfide bond between itself and the thiol. And then since these bonds are easy to break, you can just remove it. The reason this works is because even if a compound has a loose affinity for the protein, so if it's not going to like immediately attach to it, but it'll actually bind, it'll shift the equilibrium towards this favored protein because of the stability of the disulfide bond. So if the protein is not if the small molecule is not attracted to the protein at all, that disulfide bond will just be reduced and the small molecule will kind of be released. But even if it has a loose attraction to it, because of the stability of the disulfide bond, the protein will favor having this edited condition. You can break this disulfide bond and even this loosely attracted molecule can just stay there, which is really interesting. That's why they tether it down. So they kind of tie it to the protein and release that rope and then just leave the small molecule sitting there. So, and then this bottom image is just a general disulfide derived from a carboxylic acid as an example. This paper actually focused on, I have to read it, thymidylate synthasase in E. coli, so not RAS. So this paper itself wasn't focusing on targeting RAS, but the paper found that phenylsulfonamide compounds were the most successful for targeting this particular enzyme. So it doesn't always have to be a carboxylic acid derivative. The key thing is this 2-carbon spacer between the group that you change and then the disulfide. So that's like kind of this key entity that they wanted to keep, but this changed to like a benzene ring with a sulfoline group on it. So that's essentially how you're able to get these small molecules to bind to that new pocket. And then as it applies to RAS, this notation S2P just means the switch 2 pocket, that new one. It allows for the disulfide bonding and of the small molecules, I don't know why I forgot that, and for hydrophobic interactions. So you can kind of see how deep it is. And then there's the cysteine that it's able to bond to, and then there's the GDP just hanging out in there in the other pocket. So the arrows, I'm going to go to this picture before I get to the next part. The arrows indicate how the switch 2 is moved. So you can see that this is a tethering compound, and this is an electrophile. So they started trying things different in the first paper, in Chaucat's paper. So you can see how switch 2 is shifted differently based on what's bound in the pocket. So when it's bound to a tethering compound, like in compound number 6, the switch resembles like inactive KRAS. So it just looks and functions just like a GDP pound KRAS enzyme would be. But when it's bound to an electrophile, that switch 1, which is over here in red, is actually partially disordered, which is interesting because it causes a lack of coordination with the metal ion, and which is good for proper nucleotide binding, which is what kind of keeps that GDP or GTP bound. And because of this, where'd it go? It might increase the affinity for GDP over GTP. So if you disorder switch 1 enough, it won't complex the metal ion as well. GTP won't be able to bind as easily, but GDP will be favored. Another reason for this is that glycine at position 60, which was key in keeping switch 1 kind of in line, is no longer able to hydrogen bond with the gamma phosphate of GTP, which is why it prefers GDP over GTP. If you say that too much, they start sounding like the same thing. So in this paper, they actually discontinued these disulfide bond forming small molecules in favor of carbon-based electrophiles, acrylamides, and vinyl sulfonamides to form irreversible covalent cysteine bonds. So the point of the disulfide bonds was that you could remove them and just kind of leave that loosely bound small molecule there. This paper was like, we want to keep it. We want to ensure that it's not really going anywhere. But more on that later.